my slides.
Okay.
Is it visible?
Yeah, it is visible. You can start.
Okay, thank you. Myself, Ansar Mahmood from University of Poonch Rawalakot, and my title of presentation is the Fighting Plant Pathogens with Green Silver Nanoparticles, a Sustainable Antifungal Approach. So here is the introduction. Green synthesis, an environmentally friendly and sustainable method of production of nanoparticles using natural sources. The conventional methods of synthesizing metal nanoparticles often involve the use of chemicals and energy-intensive processes, contributing to environmental pollution. The green synthesis approach aligns with the global efforts to adopt eco-friendly practices, research, and applications. It utilizes natural resources such as plant extracts, microorganisms, and biocompatible agents, minimizing the reliance on harmful chemicals. The versatility of metal nanoparticles synthesized through green methods of potential applications. Their unique properties can be harnessed for diverse functions, including antioxidants, catalytic activities, and making them valuable tools in both biological and agricultural contexts. So, using the nanoparticles in agriculture, especially, as we know, there are a number of fungal diseases which harm our crops throughout the years. So, the eco-friendly approach we can use to combat this disease, because the agrochemicals which are routinely used to combat fungal disease are highly toxic and they cause environmental pollutions. So the objectives of my research was to establish the efficient eco-friendly synthesis routes for silver nanoparticle synthesis, and then utilize advanced analytical techniques like XRD, morphological and physiochemical characteristics of the nanoparticle. And the last one is to assess the antifungal activity of nanoparticles through in vitro and in field experiments. Uh, The methodology I adopted for this research, first of all, the plant extract preparation. So the plant used for the preparation of nanoparticle was the Viola odorata plant material. The whole plant was grinded into powder and made a uh, extract using distilled water, and then nanoparticle was synthesized by mixing the plant extract with silver nitrate solution under optimum conditions, and then characterization they were characterized using different techniques like using visible spectroscopy, CM, EDX, and XOD. And then antifungal activity was assessed through in vitro petri plate assay and in vitro leaflet assay using leaves of the plants. So here are the results. First of all, the synthesis of nanoparticles. We use the Viola odorata whole plant extract that was converted into powder, and then we made an extract using distilled water. And after that, the extract was mixed with silver nanoparticles, and you can see in a Eppendorf tube they are the pure silver nanoparticles that was characterized using UV visible spectroscopy, and we found a unique peak at 455 nanometer, that is the SPR of silver nanoparticles, and it showed the synthesis of silver nanoparticles in the reaction mixture. And then here is the SCM picture, SCM figure of the nanoparticles, and here we can also see the spherical silver nanoparticles with an average particle size of about 18 nanometer. And then EDX spectrum is clearly showed the synthesis of silver nanoparticles, presence of silver in the material, and X-active pattern uh, revealed the crystalline structure of nanoparticles. Here is the application of nanoparticles. We used basically that is the anthracnose disease, which is caused by Colitotrichum capsai, a fungus which cause anthracogenesis in chili plants. So we isolated that Coleotrichum capsicum from the chili plants, and then in vitro analysis showed here we use there is a control. There is 25%, 50%, 70% silver nanoparticles, and when we have used the silver nitrate solution as a control, and then fungicide that was mecnozoib was used as a positive control. So we found here we clearly seen at 75% of silver nanoparticles there is a little bit growth of capsicum Coleotrichum capsicum, and while in control the plate is full of Coleotrichum capsicum. When we use here a leaflet assay here also use on leaves, there is a symptoms of the disease on the leaves of the chili plants, and here we can see at 75% there is a very low growth of the uh, pathogen. So on these aspects we have published some research here. One of them is the green synthesis of silver nanoparticles for antifungal activity against tomato fusarium wilt caused by Fusarium oxysporum, that was published in Biocatalysis and Agriculture Biotechnology. And again, we have published green fabricated silver nanoparticles from Quercus in canna leaf extract to control the early blight of tomatoes caused by Alternaria solanae, and that was published in BMC Plant Biology. And then green senses of silver nanoparticles for their antifungal activity against anthracnose disease causing Coleotrichum capsicum, and we have published again it in Biocatalysis and Agriculture Biotechnology. So, in conclusion, the green synthesis approach aligned with global sustainability efforts. Silver nanoparticle exhibited notable antioxidant properties, showing potential oxidative stress, demonstrated strong antibacterial capabilities, hinting into applications in healthcare control, highlighted the preventive role against fungal attacks on plants, offering agricultural uses, significance in safeguarding plants from fungal infections, promoting crop health, supports sustainable farming practices with reduced reliance on chemical pesticides, encourage further research and development in understanding and expanding the use of silver nanoparticles. Thank you very much.
Thank
Hello.
you. Thank you, Dr. Ansar. This is a wonderful presentation. If you have a question for Dr. Ansar, you can drop it in the chat box and please tag his name. Our
Okay,
next,
thank you. Thank you, everyone.
thank you. Our next speaker is Muhammad Said Akhtar. Dr. Muhammad, if you are available, you can unmute yourself and start your presentation. Muhammad Said Akhtar. Are you available?
While we wait for your next presenter, I have one question for the current presenter, Dr. Ansar. Your research highlights the use of green silver nanoparticles as an antifungal solution. Could you elaborate on how this sustainable approach compares in effectiveness and safety to conventional chemical fungicides used in agriculture?
Hello.
Hello.
Dr. Ansar?
Yes.
Should I repeat the question?
Yes.
All right. So, in your research that you presented right now, you highlighted the use of green silver nanoparticles as an antifungal solution. So, would you
Yes.
elaborate that how this sustainable approach that you mentioned compares in effectiveness and safety to conventional chemical fungicides that we use in agriculture?
We actually use the green synthesized silver nanoparticles. So we do not use any harmful chemicals during the process of synthesis also. And instead of synthetic chemicals, as you know, they are also harmful to our environment. They causes the pollutions through surface runoff or any other sources. So silver nanoparticles we use in a very small concentration, as you know, 30 ppm, 50 ppm, not more than 100 ppm. So that's why they are sustainable to environment.
Thank you so much, Dr. Answer. With this in mind, that every country region is now moving forward green nanotechnology. So, for young researchers, that we have many joining us today for second day of Emerging Scientist Season 2, what do you think are the biggest challenges and opportunities in bringing eco-friendly nanoparticle research from the lab to real-world agriculture applications?
Yes, this is actually the problem. We used to test more, or we use to experiments more. Actually, we need to find the optimum conditions and optimum concentrations or optimum metal nanoparticles for against different types of pathogens. So this is actually the challenge for the researchers, for the scientists, to find out which type of nanoparticles or which concentrations or what type of concentration or what type of disease can be treated. Toxicity of metals, exactly, we know they are very toxic at certain concentrations. So their toxicity in animal